DNA is the molecule of all life on Earth. It has two main functions or purposes that make it so important. The first is that it carries all the information needed to make a single organism alive. And the second is that it is able to copy one strand of DNA and make another. The most important concept of DNA is that the information needed for self-replication is contained within the DNA itself. Before it was discovered that DNA is the molecule of life, it was thought proteins were because of their complexity and structure. But in fact, it is the simplicity of DNA and its structure that make it the molecule of life. A normal DNA strand takes the form of a double helix. It forms this way because of its chemistry, like its hydrogen bonds, bases, sugars, and phosphates. The four bases, or nucleotides, of DNA are adenine, thymine, guanine, and cytosine. Adenine always base pairs with thymine, and guanine always base pairs with cytosine. Adenine and guanine are purines, and cytosine and thymine are pyrimidines. The backbone of DNA is phosphate sugar bonds that repeat. DNA as a whole carries a negative charge, which is an important part of its chemistry that is featured later on. DNA compaction is an essential part of cell biology. To put this into perspective, take a cell that is approximately 30 micrometers in width. It also has a nucleus that is about 6 micrometers in width. This nucleus has to contain 6 feet of DNA at all times. Without compaction, the cell would not be able to handle this length of DNA and it would not survive. Compaction has two main purposes. It allows the cell to remain organized and it keeps the DNA accessible, while also preventing death. Again, the main takeaway here is that the cell will die without DNA compaction. DNA compaction begins with a double helix, its normal form. And we have to remember that DNA carries a negative charge. To begin, the cell recruits 4 binding histone proteins. They are H2A, H2B, H3, and H4. 2 of each of these proteins bind to form an octamer. Octo, because there are 8 total proteins. Each histone always binds in the same position for the octamer, and with all 8 bound, they form what looks like a spool. This frontal view only shows 4 histones. This view is from the side, and you can see how the 8 histones are put together. It will also be important for later to know that each histone has a histone tail. This tail is rich in the amino acid lysine, which is positively charged. The octamer forms so the DNA can wrap around it. Histones, like histidine, the amino acid, are positively charged. This charge is crucial because it's what attracts DNA to it. So the DNA, in its double helix form, wraps around the octamer. So if we put this a bit more to scale, the long strand of DNA wraps around the histone like rope on a spool. Here is another side view. The combination of the octamer and the DNA around it is called a nucleosome. And this process happens many times. The DNA compacted in this form is referred to as beads on a string, for obvious reasons. The DNA in between the nucleosomes is called linker DNA. The next part of compaction starts with the 5th histone protein called H1. H1 is positive as well. It binds to the linker DNA directly in front of the nucleosome. The H1 histone is known as the linker histone. It changes the natural path of DNA so it can begin coiling. This now forms the 30 nanometer fiber. The 30 nanometer fiber is the last part of DNA compaction that scientists are sure of. But the DNA compacts more and forms a chromosome, and the chromosome is the last form of DNA compaction. DNA compaction, as we learned, is essential to the cell's survival. But the cell does not always keep the DNA as compact as possible. For instance, during replication, it is not as compact, and when the DNA needs to be repaired, it is not completely compact. To understand how the cell gets access to DNA when needed, we first have to learn about lysine. Remember, lysine is an amino acid that carries a positive charge naturally and is the main amino acid in histone tails. The chemistry of lysine is what locks the nucleosomes in place during compaction. This is due to the attraction of the negatively charged DNA and the positive lysine. So during normal compaction, lysine's charge is present and the nucleosomes are locked in place to keep the DNA compact. For access to DNA, histone acetyltransferases are used, or HATs. These enzymes acetylate the lysine and neutralize the positive charge. Without lysine's positive charge, it doesn't have the attraction to negatively charged DNA anymore. This results in the nucleosome loosening and the compaction weakening. This is how the DNA becomes exposed and the cell can do what it needs to do. Once the cell no longer has a need for the DNA, it wants to compact it again. So this is where histone deacetylation complexes come in, or HDACs. These enzyme complexes reverse the effects of the HATs. They deacetylate and restore lysine's positive charge, therefore restoring their attraction to DNA. This locks nucleosomes in place and restores the DNA compaction. Another way cells gain access to DNA sequences is chromatin remodeling complexes. They use energy from ATP hydrolysis to physically force nucleosomes out of the way. While they move the nucleosome, they also thread adjacent DNA onto the nucleosome. These complexes create short regions of DNA that are freed up from compaction and are accessible. DNA replication is when one DNA molecule is copied, resulting in two new DNA molecules. DNA replication is made possible because of base pairing. The bases have an attraction to one another that makes replication possible. And remember, adenine always pairs with thymine, and guanine always pairs with cytosine. This is how the information for DNA replication is in the DNA itself. The method of DNA replication is called semi-conservative replication. The old strand from the original DNA serves as a template for making the newly synthesized strand. So as a result, each DNA molecule has an old strand and a new strand. Replication begins with DNA as a double helix. There are approximately 3.2 billion bases in a DNA molecule, so replication must occur very quickly to remain efficient. This is achieved using origins of replication. There are about 10,000 of these specific sequences of DNA along the molecule. So replication happens along these origins simultaneously. It is important to know that DNA is 5' to 3'. DNA replication can only progress in the 5' to 3' direction, and the replication fork is bidirectional, so both strands are made 5' to 3', but they occur in opposite directions. The first strand is called the leading strand, and the second strand is called the lagging strand. So replication begins when an initiator protein binds to an origin of replication. Then DNA helicase, an enzyme, binds to the origin and pulls the strands apart by disrupting the hydrogen bonds that keep the bases together. It's like the molecule is being unzipped. The Y shape is called the replication fork, and it is the site of active DNA replication. So these two single strands will each serve as a template for making two new strands. Single-stranded binding proteins bind to each strand near helicase to keep the single-stranded DNA from reassociating with its complementary strand. The leading strand is easier for the cell to replicate, and to make it easier to see, the new strand including the bases is blue, and the old strand including the bases is black. Firstly, a primase binds to the 3' end of the strand and puts a small segment of RNA down. The RNA is in red. These primers are generated by DNA primase and are essential to DNA replication, because DNA polymerase cannot start replication on its own. The leading strand only needs one RNA primer. Next, DNA polymerase binds nearest to the replication fork directly next to the RNA primer. To keep DNA polymerase bound to the template strand, a clamp protein encircles the molecule and makes sure it does not float away. An additional protein called the clamp loader loads the clamp protein onto the DNA strand, but once it does its job, it floats away. DNA polymerase is what takes the complementary base and pairs it with the one on the strand. It binds them by creating a phosphodiester bond. Individual nucleotides enter the reaction site of DNA polymerase as deoxynucleotide triphosphates, each specific to the base it's pairing to. For this strand, DNA polymerase is moving continuously toward the replication fork in the 5' to 3' direction. The lagging strand is more complicated. This is because the strand is backwards. DNA is made 5' to 3', but the template strand is not in that direction. So the new strand needs to be made in segments called Okazaki fragments. And you have to imagine that the leading strand above is being made continuously at the same time. And helicase and the single binding proteins are not cut out, but they are moving as well. So first, primase binds to the 3' end of the template strand and lays down a segment of RNA primer. DNA polymerase binds to the template strand along with a clamp protein helped by the loader and begins synthesizing. But it has to work backwards, moving away from the replication fork. Once DNA polymerase has made a fragment, the clamp protein releases the DNA polymerase and it floats away. Then the enzyme exonuclease lands on the 3' and chews up the RNA primer on the fragment in front of it. Then another DNA polymerase puts down new DNA in place of the just chewed RNA. Next, DNA ligase bridges the gap between the Okazaki fragments. This process repeats to form the new strand. The primase puts down RNA primer. DNA polymerase finds the template strand along with a clamp protein and the loader. They make a new strand of DNA, or a fragment, and then exonuclease chews up the RNA primer. Repair polymerase puts down more DNA, and then DNA ligase comes in to repair the gaps. During replication, DNA proofreading occurs. This is when DNA polymerases check their work regarding the bases they added. When an incorrect base has been paired, DNA polymerase recognizes it mainly because the structure is not smooth like it should be. Polymerase then uses 3' to 5' exonuclease to remove the incorrect nucleotide. Then the process reverts back to normal and the polymerase continues to make DNA. After replication is done, there is something called the end replication problem. Since DNA polymerases can only synthesize DNA from 5' to 3', there is a problem with the lagging strand. It is synthesized in the opposite direction using an RNA primer as a starting point. So by the end of replication, there is a short segment that cannot be replicated. Left this way, the chromosome will shorten with each replication the cell does. Telomeres are the solution to this problem. At the end of chromosomes are telomeres with a guanine-rich sequence. So telomerase recognizes this sequence. And with its own RNA template strand, telomerase elongates the template strand in the 5' to 3' direction and adds additional repeats as it moves down. The lagging strand is then completed by DNA polymerase alpha, which carries an RNA primase as one of its subunits. Now, the DNA is copied with no information being lost. Damage to DNA is randomly occurring in cells constantly, and this damage can be single-stranded or double-stranded. Single-stranded damage has three main methods depending on the type of damage. They are mismatch repair, base excision repair, and nucleotide excision repair. For double-stranded breaks, the methods are homologous recombination or non-homologous recombination. Mismatch repair is dedicated to fixing mistakes that sneak through DNA polymerase proofreading. This mechanism fixes about 99% of the mistakes that DNA polymerase makes, which are few to start with. It occurs right after replication. In prokaryotic cells, they recognize the new strand versus the old strand because of methylation. They methylate their old strands so that they can tell the difference. But in humans, it's unknown how the cells know which strand is new and which strand is old. And to begin mismatch repair, a beta complex made from MMR proteins recognizes and binds to the incorrect base. Then, an alpha complex, also made of MMR proteins, comes in and cuts the incorrect base out along with some adjacent bases. Then, DNA polymerase is recruited and replaces the patch of DNA with the correct sequence. And lastly, DNA ligase comes in and seals the gaps. Base excision repair is used when only one base is mutated. An example of a single base mutation is deamination. This can convert cytosine to uracil, and uracil does not belong in a DNA molecule. So first, the modified base is detected by glycosylase. Then it is cut out of DNA individually by glycosylase. This is different from mismatch repair because only one mutant base is being removed, rather than other bases adjacent to it being removed as well. This leaves a small hole in the DNA. The single nucleotide gap is then filled in by DNA polymerase, and finally, DNA ligase seals it in place, completing the molecule. Nucleotide excision repair is for repairing damage to major distortions of the structure of DNA. It starts with scanning proteins going along the DNA structure looking for distortions. Remember, DNA is a smooth structure, and any distortion will be easy to recognize by the scanning proteins. When a distortion is found, the double-stranded DNA in that region is separated and kept separate. Then, the DNA is severed on that strand by an exonuclease on either side of the damage, leaving a gap in the DNA. Then, the gap is filled in using an RNA primer and DNA polymerase. It is then sealed in place by DNA ligase. Double-stranded breaks in DNA completely sever the DNA molecule into two pieces. These are the two main mechanisms for double-stranded break repair: homologous recombination and non-homologous recombination. Homologous recombination is better for the cell, but it is not always used. This method requires homologous pairs of DNA, and during compaction, the homologous pairs are not always next to each other. To start, a nuclease chews back one of the two strands of DNA at the double-stranded break. Then, the single-stranded broken end of DNA invades the nearby DNA molecule, creating the D loop. This creates a branch point where two DNA strands, one from each molecule, cross. The invading strand is then elongated by a repair polymerase. As the DNA is synthesized, helicase is used just like a replication fork to allow additional DNA to be made. It is completed by nicking the invading strands, allowing them to return to their normal positions, and sealing the nicks with DNA ligase. This leaves two completely intact DNA molecules. Non-homologous repair is a bit simpler. It starts with the double-stranded break of the DNA molecule. The cell takes the two ends of the molecule and blunts them. This removes nucleotides so the ends are now symmetric. This is why non-homologous repair is not favorable. DNA information is lost in the blunting process, and there is a chance that what is lost is vital DNA. Vital DNA can code for proteins that are essential to a cell's life, so this method is very risky. The pre-initiation complex is assembled because RNA polymerase cannot initiate transcription on its own. General transcription factors are needed to start the process. So let's take a DNA molecule ready to be transcribed. On this molecule, there is something called a TATA box. This is a repetitive sequence of thymines and adenines. This is a portion of the DNA that is easy to pull apart and acts as the promoter for the first transcription factor to bind. First, transcription factor 2D, which also contains TATA binding protein, binds to the TATA box. Then, transcription factor 2A binds to TF2D. Then, transcription factor 2B is recruited and binds to both A and D. At this point, RNA polymerase II comes in and binds behind the TATA box, escorted by transcription factor 2F. And this polymerase has a carboxyl tail domain, or CTD. Next, transcription factor 2E binds to TF2D. This weakens D's affinity to the DNA so it can move. The last transcription factor is TF2H, and it binds to RNA polymerase and moves it. After all the transcription factors have bound, phosphorylation occurs, and E shifts D up and out so RNA polymerase II can move forward and transcribe. Within the RNA polymerase, the DNA has separated and RNA is being made and dangles out of the RNA polymerase. Once RNA polymerase reaches a stop codon, phosphorylation signals RNA polymerases to release from the promoter. Eukaryotic cells have the opportunity to inspect their mRNAs before translation. As part of this, mRNA is processed. Additions to its beginning and end are the cell's way of showing it is ready for translation. Only mRNAs with these additions can leave the nucleus. The first part of mRNA processing is when mRNAs receive a 5' cap. This is a specifically altered nucleotide base added to the 5' end of RNA. It marks the beginning of the mRNA as genuine. The second part is when mRNAs receive a poly-A tail. This tail is usually hundreds of adenines long, added to the mRNA's 3' end. It marks the end of the mRNA as genuine. These two modifications result in the mRNA being more stable and allow it to exit the nucleus. Proteins that are recruited to the 5' cap and the poly-A tail interact and cause the good, genuine mRNA to circularize. This tells the cell that it's dealing with a good, fully processed mRNA. The third part of mRNA processing is splicing. Genes have their protein coding sequences interrupted regularly by long, non-coding intervening sequences called introns. The actual coding sequences are exons. Each intron has an important conserved sequence at its ends and somewhere in its middle. These sequences tell a cell they need to be removed. Using the sequences as a guide, a complex protein machine called a spliceosome is used. The spliceosome is made of RNA, and it removes introns and ligates exons together. Splicing begins with the mRNA. An intron is in between two exons and it needs to be removed. The intron has a 5' splice site with a guanine, a branch point with an adenine, and a 3' splice site with an adenine. The mRNA is spliced at the 5' site, and it folds over and attaches to the branch point. This forms a lariat. Then it is removed completely through splicing at the 3' end of the intron. Then the exons are ligated back together. Alternative splicing is when not all the adjacent exons are used. This results in the mRNA encoding for different proteins than we have genes in our DNA. So if you have an mRNA with these exons and introns in between, some exons are deleted to make new proteins with different functions. There can be many results giving rise to different proteins with different shapes and different function. Translation is the act of an mRNA being coded and a protein resulting. It occurs once mRNAs have been processed and have left the nucleus. Before we get to the act of translation, we first have to understand the different elements that make it work. So there will be a lot of explaining, and then we will jump into translation with all the elements. First, we need to know what the RNA is coding for. So sequences are decoded in sets of 3 nucleotides. These sets of 3 nucleotides are called triplets or codons. And in order for us to know what amino acids come from each codon, we use a genetic code. And keep in mind, there are only 20 essential amino acids, but only 64 codons. This is an important aspect of cellular composition because it allows the cells to have more than one codon represent a single amino acid, and the cell has a buffer against mutations. This is the genetic code showing the different codons for different amino acids. Reading frames are a sequence of DNA that represent a protein, and the reading frame begins with the start codon, AUG, and ends with the stop codon. There is only one start codon, but there are 3 different stop codons, UAG, UAA, and UGA. But it is important to know that mRNA do not bind directly to their corresponding amino acids. They need something to bridge the mRNA and the amino acid, and that bridge is tRNA. tRNA have a 3-base anticodon on one end. This codon is meant to directly base pair with the codon on the mRNA. This method provides recognition of the mRNA. On the other end of the tRNA is the corresponding amino acid via charging. But we also only have about 31 tRNAs coding for the 61 different codons. So how can 31 tRNAs encode for 61 codons? The answer is wobbling. Wobbling of base pairs allows one tRNA to recognize multiple codons. It usually occurs in the 3rd position of the codon. So if we have a tRNA with a GAG anticodon and leucine on the other end, the mRNA codon should be CUC. But due to wobbling, the anticodon GAG can also base pair with CUU. This is how the 31 tRNAs can code for 61 different amino acid codons. So how do the tRNAs know which codon goes to which amino acid? Well, they use tRNA synthetases. These enzymes can just look at an anticodon the tRNA has and immediately link the appropriate amino acid onto the tRNA. The ribosome is a large conglomeration of RNA and protein molecules that come together to form a complex. The complex is made of a large subunit and a small subunit. The larger subunit forms the peptide bonds between amino acids. The small subunit matches mRNA codons to their proper tRNA anticodons. In the larger subunit, there are 3 sites or voids. Each is shaped differently for a specific purpose. The first site is the A site, or amino acyl tRNA. This site carries a tRNA with a single amino acid. The second site is the P site, or peptidyl tRNA. This site carries a peptide chain of amino acids. The last site is the E site, standing for empty. This site should always remain empty of amino acids and tRNA. So now that we know the components, we can start to put them together. The cell begins translation with initiation. So we have an mRNA strand that is ready for translation. To start, an initiator tRNA associates with a free small ribosomal unit. Then, the small subunit binds to the 5' end of mRNA. The small subunit keeps the mRNA engaged while the initiator tRNA scans for an AUG start codon. When the AUG is found, the initiator tRNA stably base pairs to it. Next, the large ribosomal subunit joins the complex and associates so that the initiator tRNA carrying methionine is in the P site. The initiator tRNA is the only tRNA that can enter the ribosome P site directly. Finding the AUG established the correct reading frame. Eukaryotic initiation factors bind to the 5' cap of the mRNA and to the small ribosomal unit. These factors recruit all of the necessary players for translation and position them correctly. So once the initiation complex is formed, elongation starts. Another tRNA with the correct anticodon and corresponding amino acid binds to the mRNA in the A site of the ribosome. The amino acid in the P site creates a peptide bond with the amino acid in the A site. At this point, we have a tRNA with 2 amino acids in the A site and a tRNA with no amino acids in the P site. This does not follow the rules of the large ribosomal unit, so translocation occurs. The entire ribosome moves 3 codons down, so the tRNA with no amino acid is now in the E site, and the tRNA with the polypeptide chain is now in the P site. Then, the empty tRNA leaves the ribosome at the same time that a new tRNA binds in the A site. Now we are following the rules. It is also crucial to know that the large subunit can only hold 2 tRNAs at one time. This is why one tRNA leaves the E site while another enters the A site. This process happens all the way down the mRNA. Termination is when the finished polypeptide chain is released to go do what it was made for. It begins with any of the stop codons mentioned before, UAG, UAA, or UGA. Once translation is complete and a stop codon is positioned under the A site, proteins called release factors enter the A site and bind to it. There are no tRNAs which recognize the stop codons. Release factors catalyze the release of the polypeptide amino acid chain from the tRNA in the P site, freeing it from the ribosome. The 2 subunits of the ribosome then dissociate and the mRNA is released. The ribosomal units and mRNA can repeat this translation. The newly made protein folds into its native 3D shape and goes off to do the job it was made for. Combinatorial control is a group of regulatory proteins working together to precisely control the expression of a gene. In our cells, one gene is usually controlled by dozens of regulatory proteins, both repressors and activators. Each of these regulators is usually transmitting its own individual signal on the time, amount, and the stimuli, both internally or externally. The end result of this is that gene expression is given a committee decision. This type of regulation is very complex but also allows true fine-tuning of gene expression. This occurs using enhancers that lie many bases away from their target gene. So we have this strand of DNA with multiple enhancers spaced out from one another. Different transcription factors, also known as regulatory proteins, will bind to the enhancers. This creates the looping out of DNA called spacer DNA. This allows the enhancer DNA to become adjacent to one another, and they can all send signals to either be activated or repressed. At the promoter is RNA polymerase II, as well as other transcription factors that are not involved with combinatorial control. Now this is where mediator comes in. It's a large protein complex that acts as a relay complex. It sits on top of RNA polymerase II and receives all the signals affecting a single gene. Those signals can be positive or negative. It receives these signals and passes them on to RNA polymerase II, telling it what genes to transcribe or not. So if 3 transcription factors send a signal for the gene to be on, and the 4th sends a signal for the gene to be off, mediator takes these votes and tells RNA polymerase II that the gene needs to be on. RNAi is found in most eukaryotes and is probably the most potent form of regulation known. RNAi is triggered by double-stranded RNA that is fully base paired. Double-stranded RNA molecules are cut into 21 to 23 base pair fragments by an enzyme called dicer. These small double-stranded RNA fragments are called small interfering RNA. siRNAs are then bound to an RNA-induced silencing complex called RISC. RISC pulls the double-stranded RNA apart. Once recognized, RISC destroys these mRNAs. Then, it uses one of the 2 strands to recognize other RNA molecules, either single-stranded or double-stranded. Less than 50 small interfering RNAs can silence thousands of pre-existing mRNAs. The reason for this amplification of small interfering RNAs is because of RNA-dependent RNA polymerase. After RISC cuts an mRNA, 2 unstable fragments are left. RDRP appears to use one of these fragments as template to generate double-stranded RNA. The double-stranded RNA is then used by dicer, generating more siRNAs for RISC so the process can continue. Humans do not have RDRP, so we don't amplify RNAi. You also should know that RDRP does not need a primer like DNA polymerase.